Okay. All right. So, I think I got this.
Let me give you some advice. The process of doing your second draw.
Okay. Finally. Looks like I'm live. I am gonna do a couple of adjustments here, but I am so happy to be here. I'm Mr. W, and I'm wondering if anybody is there. I'm not seeing anybody in the chat so far. Why don't you just introduce yourself and say hello? Just give me a I'm here, and that'll be really thrilling. Alrighty, so I see a couple of folks there. Fantastic. Maybe I need to do a little bit of work here on my other little monitor here. Okay, fantastic. So Yuna's here again, and I've got a whole bunch of folks. And let's see if I can get this stream to show up over here too. And let's activate that. Okay, fantastic. And let me just do a couple of other things here. Oh, fantastic. I'm seeing a lot of people signing on. That is really great. So, thank you so much for joining me for this stream. I am so sorry for the delay. I'm just going to try and adjust a couple of things here on my monitor so I can get things 100% set up to go. And let's see what we got here. Fantastic. Okay, making some progress here. Okay, all right. So now I can see the chat. I see some folks from Berkeley High School here. I see my hero, Mr. Glimmy. Thanks so much. I need to be a bit louder, and let me just set that up a little bit. So, how's that going? All right. Really hard to hear. And just lost my second. Oh, that's a bad thing. Okay, so I think I need to do this. Wasn't really what I was expecting. Okay. All right, Mr. Glimmy, that's fantastic. All right, Mr. White, thank you so much for showing up. This is really fantastic. Okay, so guys, you ready to do a little singing? Because I thought we'd start with a song just to get things moving a little bit here. So if you guys are ready, I thought we'd sing a little bit of glycolysis. Give me a thumbs up or something if you're ready to do a little bit of singing. Anybody there? Anybody ready? Want to hear a little glycolysis? Okay, now here's a question. Maybe people can give me some feedback. Can you hear the music? Somebody give me a yes if you can hear the music from glycolysis. Somebody type in the chat. Can you hear any music? Give me a yes or a no. Anybody hearing any music from my monitor? Nothing coming through? All right. So, can't tell if actually that audio is going out to you folks or not. So I'm wondering if somebody could just type in the chat as to whether or not you're hearing me or not. No, you can't hear that. That is super disappointing. So that's something that I think I'm going to have to figure out. All right, so I'm hearing from a lot of folks that you can't hear that stuff that's coming through. What a bummer. I'm wondering if there's any way around that. Okay, how about this? I don't know if this is going to work. Let's try it.
Hey, sugar. Come on over here, sweetie.
Can you guys hear that?
Let's break it down.
It's from my phone. It's pretty lame. Anybody hear that coming through? Oh, fantastic. More and more of my students are coming on. You know what I think we're going to do? I think we're going to skip some of the musical extravaganza that I thought was going to happen. And I think instead what we're going to do is we're going to go ahead and I'm just going to, all right, you could hear that. Okay, Israel, you could hear that? Let's try it. Okay. Now the question is, can I get these things to coincide? I don't think I can. Let's see.
Hey, sugar. Come on over here, sweetie. Let's break it
All right, let's
down.
sing it. Glycolysis, reactions, enzymatic actions, energy transactions. Takes glucose and molecules of sugary, breaks it down for NADH and ATP. It's an anaerobic cytoplasmic pathway that amazes, organized easily into 3 phases: investment, cleavage, and energy harvest. Tell me later which one you like best. Investment, activation, energy supplied. Cleavage, our 6 carbon sugar divides. Harvest, we get our energy yield. So beautiful, so intricate, keep your eyes peeled. Glycolysis. Come on, sugar for the breakdown. For the breakdown. Investments, like striking a match. That energy you put in makes the fire catch. For glycolysis investments, 2 ATPs, which act as activation energy. Enzymes take phosphates from ATP, jam them onto glucose, rearranging it to fructose, leaving fructose 1,6-bisphosphate on the stable. With 2 phosphates, it's highly unstable. Moving us to the second phase, the cleaving of fructose bisphosphate. Because glycolysis means splitting sugar, you can see. Cleavage yields 2 molecules of carbons 3. One is glyceraldehyde 3-phosphate G3P. It continues on our pathway broken down for energy, but a second one, an enzyme, will immediately convert into a second G3P glycolysis. Come on, sugar for the breakdown. For the breakdown. Phase 3, G3P gets rearranged and oxidized by an enzymatic assembly line that harvests energy from each G3P, 1 NADH and 2 ATPs. Double this yield per G3P to 2 NADH and 4 ATPs. That's the gross yield. For every glucose in, a generous accounting of glycolysis is win, but 2 ATPs were invested in phase 1, so you net just 2 you can use to jump a run. Put 2 in, get 4 out, your net gain is 2 ATPs that you can use. If that doesn't sound very much, it's cause it ain't. There's tons of energy left in pyruvate. The 2, 3 carbon molecules were left without the end, and what happens to pyruvate is going to depend on the metabolic pathway where pyruvate gets sent. If it's anaerobic, it'll be fermented, but in aerobic cells, pyruvate's termination will be the Krebs cycle and total oxidation glycolysis. Come on, sugar for the breakdown. For the breakdown. Glycolysis starts with investment of 2 ATPs to the glucose that we started with. The product is cleaved into 2 G3Ps from which the cell harvests NADH and ATPs. This anaerobic pathway is respiration first phase, billions of years old, evolved in ancient days before O2 accumulated in the seas, before eukaryotic cells arrived on the scene. It's everywhere, ubiquitous in every organism. Bacteria, sequoia chi, no matter your metabolism, happens in the cytoplasm, doesn't need no organelles. Want to find glycolysis? Look in any cell. Sing it with me. Come on, sugar for the breakdown. For the breakdown. Glycolysis. Come on, sugar for the breakdown. For the breakdown. Oh, yeah. All right. So, folks. Oh, that was awesome. Okay, so that's it. That's how we start. And now let's get into talking about this year's AP Bio exam. All right. So to begin with, let me just move things around here. So I'm looking at what I want to look at. Okay, beautiful. So first things first. Everybody knows, really different AP exam this year. Forget about multiple choice questions. There's just 2 FRQs. You're going to take them at home, and it's open book, and the whole thing, 45 minutes. Now, the first question is going to be a long question. It'll be some scenario with a lot of data in a table or a graph, and you'll have to analyze that, and you'll have to describe and explain the underlying biology. You'll have to identify experimental design procedures, what's the control group, what's the experimental group, dependent variable, independent variable. You'll have to analyze data. There's going to be some kind of calculation, and then you're going to have to make and justify a prediction. I want to emphasize that justification of predictions, that's really the most important thing that you do on these essays. So make a prediction, justify it, and we're going to practice that in just a minute. Now, the second question will be shorter, and there'll be some scenario describing a biological phenomenon that gets somehow disrupted. I'm going to give you an example of that in the practice that we're going to do right now. And you'll have to describe and explain biological concepts connected to that scenario or describe the characteristics of that. And then you'll predict the causes or effects of a change in this system and then justify. And again, I don't think it's any coincidence that justify is showing up in both of these. It's claim, evidence, reasoning. That's what it's all about. I can't tell you exactly how long the first question is going to take, but it's 45 minutes. I think it's going to break down about 2/3 versus 1/3, but I can get that information, and I just heard from somebody, one of my students, who's reporting that it's 25 minutes. And someone's asking me if this if this lecture is going to be recorded and posted later, and I'm going to do one or two things. If the quality of this is good enough, I'll post it as is. If not, I'm going to make separate videos about this and then post that. So let's go on. So let's talk about how you can set yourself up for a 4 or a 5. Now, if you're one of my students at Berkeley High School, you're already set, okay? If you're working with another teacher who's using sciencemusicvideos.com, you're also already set. But if you're not set, because you're not in one of those two categories, then here's what you do. Here's a screenshot of my website, and this is my AP Bio exam review system called Biomania. So if you go ahead and you click on this link over here, it'll take you to this page. And you see over here, this gives you two important things. This first link over here is a plan that'll walk you through the next couple of weeks to get you ready for the AP exam. And then there's a practice journal, and the practice journal looks like this. Before I do that, someone's asking me a question about how many points for each question. There were point values that were attached, and you can find these online, and I can share them with you all later. I think basically 2/3, 1/3 is the way to think of it. So 2/3 of the credit that you're going to get is going to be on that first question that I was talking about, and another third on the second question, okay? So if you click on this link over here, your Biomania FRQ practice journal, you're going to get to this document. And it's a Google Doc. It'll automatically make a copy of it. And I just want to walk you through what's going on here. These here are some introductory videos that sort of guide you through how to do flashcards, how to do multiple choice questions, and how to do FRQs. Mostly, you're obviously going to be focusing on the FRQs and the flashcards, because those things are really in line with this year's exam. But I would strongly suggest that you work the multiple choice questions also, just because they're going to help you master visual representations. They're going to give you the kind of feedback that you're going to need that's really going to lead you to a very high level of content mastery. So don't skip those. So these are some introductory videos that are telling you about that. This is a link to one of the most important things that you have access to. The College Board publishes their FRQs online with scoring guides. I'm going to show you that in a second. You should be practicing with all of the relevant FRQs, the ones that relate to units 1 through 6, and writing those. In fact, writing those multiple times. That's where you're going to see the kind of challenges that you're going to face on May 18th. That's the new date of the exam. Someone's asking me how you access that document. I'm just going to jump back. So here you click Biomania, AP Exam Review, and then there are 3 links, And this one over here is a link to that Google Doc, okay? So I think we got everybody's questions there, and now let's go on. Okay, so this is giving you a week-by-week schedule. If you haven't started yet, we've got till May 18th. There's plenty of time to get this, plenty of time to get this right. And what I've done is I've linked between, for example, week 1 and then the writing that you should be doing for week 1. And the big idea is this. I know that I'm doing a live streaming video, but now is not the time to be watching a ton of videos unless they're directly in line with practice. Now is not the time to be rereading your textbook. Now it's the time to be doing what's called retrieval practice. And retrieval practice is when you're posed with a question and you try and reach into your mind and think about what you can say, because that's how you solidify memory. That's how you make connections between different ideas. So that's what you've got to be doing. And I'm seeing a lot of people come on. I just want to say hi and thank you to all the people who are coming on. But spend your time doing the College Board reviews, and also spend your time doing the reviews that I've set up for you on my Biomania AP Bio review system. And here's the college, this is AP Central. And if you just Google past AP Bio exams, you're going to get to this page. This link will get you to the actual questions. This link will get you to scoring guides. You even have sample responses. You probably don't want to worry too much about the Chief Reader Report, but you can read that too because it'll talk about strengths and weaknesses. So here is one of the things that I'm offering you on my website. So this is, again, from the Biomania page. There's a link that takes you to multiple choice questions, practice FRQs, and flashcards. These are the practice FRQs. And so what you do is you just select the units that you want to study, and Biomania will pose these questions to you, and you answer the question, and then you see the sample answer that I've put together. And that is the best way to study. You should be spending hours doing that. And I also want to point out that you can get this same content as part of a phone app, all right? And the download's for free. It's on the Apple Store. Soon it's going to be on the Android Store. And basically, what you can do is you can unlock all the content for $9.99. It's the best bargain in biology education, and I totally want to encourage you to go ahead and do that. So do a lot of practice on Biomania, and we're going to do that right now. Oh, and this is so sad because you're not going to be able to hear this. I'm going to fix this so you can hear this beautiful song that I put together with my music producer, Max Cowan. He runs a band that's called Attikid, one of the coolest funk bands in the Bay Area, possibly in the United States and possibly in the world. Anyhow, we'll get that music to you soon, but that's not going to happen today. But you did see the cool animation. So anyhow, what I want you to do is I want to see some writing. And I want that writing to go into two places. One is if you've got your journal set up and ready to go. If you're my student and you have your journal set up and ready to go, you can do some writing right now. And this can be, I don't know if it's probably your week 2 writing, but it doesn't matter. We're going to walk through a couple of questions together on the AP Bio, through the AP Bio Biomania app. Now, what I also want to do is this. I want you to be putting answers into the chat, okay? So the chat is going to be your way of giving me feedback. If you're my student, you know that I'm a pretty, I talk a lot for sure, but I'm also a very interactive teacher. And it's hard for me to teach without getting feedback. And here, all I got is the chat. So when I ask questions, what I want you to do is put answers into the chat, and I'll monitor that to see how people are doing. So let's start, okay? So here's the first question, and just not to make this incredibly difficult or tedious, I know that it's hard to read on a screen like this in a PowerPoint presentation, so I'm going to read these out loud. I know that's not necessarily the best practice. Some of my colleagues are probably thinking, where's all my cognitive science? But I just want to also show you something. I'm going to go back a little bit. Okay, so if you go to Science Music Videos and you click on this link over here, live question of the day, I put the text for all of these questions there. So you can keep the text up and you can continue to look at it. I wasn't able to put the images that'll be up, but you'll see those images. So just note that you can see the images right there. And I saw somebody point out that the app is available for Android too, and that's fantastic, though I don't know if we have exactly the latest version. So anyhow, that's the deal. So let's go on. I'm going to go back to where we were. All right, so I'm going to read this aloud. You can, again, read it on that live page on sciencemusicvideos.com. If it's comfortable for you, you can read it here. I was originally going to provide some music over here, but that ain't working, so that's not going to work. So I'm just going to read this. So here's the question. This is a Unit 6 question from my Biomania app. Proto-oncogenes, when they mutate to become oncogenes, can cause cells to become cancerous. A lysogenic virus infects a particular cell type and integrates into its genome into a site that contains a proto-oncogene. This transforms the cell and increases the level of protein X. Protein X, in turn, increases cellular proliferation. That's the division of spread, and that's really what cancer is. Tumor suppressor genes code for proteins that prevent cells from becoming cancerous. A compound P is known to increase the level of tumor suppressor proteins in that cell type. A second compound, Q, helps in stimulating protein Z. Protein Z has been shown to be capable of binding to X and rendering it inactive. In the graphs at right, a minus sign indicates that substance P or Q was not applied to experimental cell cultures, while a plus sign indicates that substance P or Q was applied. And here's your job. You're going to identify which of the following graphs correctly represents the mode of action of P and Q and justify your response. So this is a lot of stuff, and I'm going to walk you through it. For some of you, I'm seeing some of you are already answering, and that's fantastic. And some of you might need a little bit more support, and I'm going to go and do that. So here's the deal. In these AP Bio questions, critical reading is the key. So here's the text that is in the introduction. And in general, in the College Board, they don't hold back on their reading level. It's at a complex reading level. So here's what I want you to do. I've got 4 questions about protein X, compound P, compound Z, compound W. So don't get ahead of me here. What I'd like you to do is determine each substance's effect on cancer cell proliferation. Does it increase or decrease it? So type in the chat right now what protein X does. Does it increase or decrease? Go. Let me see something show up in the chat. Protein X. We're just doing protein X right now. Increase or decrease? Want to see some responses? I'm going to have a sip of coffee while you do that. Fantastic. I'm seeing a lot of people say increase, and that indeed is correct. Now, how do you know that? Well, protein X increases cellular proliferation. So it's all about critical reading. Now, how about compound P? What does compound P do? Please write your answer to compound P. Put it in the chat so I know how you're doing. Compound P. All right. I'm seeing so many great answers. Sky, Jojo, Lizzie, Alan, Grant, Chaticus, Melissa, Amanda. All right. Now I'm seeing oh, this is awesome. You guys are getting it. All right. So we're decreasing, and that's exactly what compound P does. Now, how do you know that? Because compound P increases the level of tumor suppressor genes, and tumor suppressor genes suppress tumors. That's their name, tells you what they do. Now let's do protein Z. All right, everybody ready? Let's start typing protein Z. What's its effect on cell proliferation? Let's see it. Let's go. Probably shouldn't clap. All right. Protein Z. Let's see some answers come in. Good, Amanda. Fantastic. Isabella, Sky. Fantastic. Let's keep those answers coming in. Want a couple of more? All right. Lizzie, fantastic. Okay, great. All right, so now, protein Z indeed decreases it. Well, how do we know that? Because protein Z inhibits, it binds with X. X is our oncogene, is moving our cancer forward. So if this is inhibiting X, then we are decreasing the amount of cell proliferation. And now we're going to go for protein compound Q. Let's see it. All right, so let me see compound Q. I'm seeing answers coming in. Fantastic. Compound Q. All right, and excellent. You guys are totally doing great on this. Compound Q is also decreasing it, okay? So we figured out our basic thing. Now, I think many of you are probably ahead of the game here already, and some of you actually put the answer in before I'd even walked you through this. That's the quality of students that I get to work with. It's such a fantastic thing. So this just, this was just for reference, right? Because, who would just be able to hold that in their head? Not me, for sure. But now what I want you to do is identify which of the following graphs correctly represents the mode of action of P and Q. And here's how this graph works. This means that in this cell culture, P was not present and Q was not present. In this one, P was present, Q was not. You get it? So which of these is right? Go ahead. Is it A, B, C, or D? Which represents the mode of action of compound P and compound Q? Let's see it. I'm seeing answers come in. I saw one. Fantastic. Let's see some more. Just see one answer so far. Everybody's thinking about this. Everybody's thinking. Fantastic. I'm seeing some more answers coming in. Got two so far. Jojo and Ariana. So great to have you guys here. Amelia, fantastic. Chadicus. Sarita, fantastic. Amanda, Nisa, Amazing Arts, Melissa, Mesty, Lizzie. Haha, this is making me so happy. Okay, so the answer, let's take a look at the answer, all right? Let's take a look at A, all right? Basically, A, we're seeing these two not here, cell proliferation is high. These two both here, cell proliferation is high. In other words, A is basically arguing that none of these substances do anything. That's unlikely because we've already seen that these substances have a certain effect. So we're getting rid of A. B, well, okay, if both of these are absent, then nothing's happening. Okay, cell proliferation is high. If one of them is present, in this case, if P is present, then we have a little bit of a decrease. And if Q is present, then we have a bit of a decrease. But when both of them are together, also high, doesn't make any sense, right? And now, finally, if we've got P and Q, we've got this one over here. So this is saying neither of them are present, all right? High. One of them is present. Lower, both of them together. This is the one that makes sense. So your job in this was to actually pull this all together and write a justification. So I want to model for you what a justification is like. So let's just walk through this together. And what I want you to do is I want you to think of words that would make sense in these lines. And basically what we're going to do is we're going to construct your justification together. Okay, so let's go. P blanks tumor suppressor proteins, which would blank cell proliferation. So, think of words that would make sense for this over here. All right, let me see you type that in the chat, please. I'm doing what I do in class. I often rub my chin as I'm thinking, but now I'm just waiting for your answers. People just rocked it on the last one, so everyone's going to do great on this. Let's see some terms like increase, decrease, you know, something like that. Go ahead. Okay, okay. Seeing some answers come in. Not going to show them yet. I've got Amazing Arts. I've got Lizzie, Jojo. Okay, everyone's doing the first one. All right, so let me show you. All right, P increases tumor suppressor proteins. We've already established that, which would, in this case, increases tumor suppressor proteins, which would decrease cell proliferation because tumor suppressor proteins suppress cancer. Now let's look at Q. Q indirectly activates X because X, what does X do? Well, people are putting in all these. You know what? I like what Amanda just did. People are putting a string of all of these answers right here. And I think that makes more sense because it's going to make this experience less fragmented. So what I want you to do is do like, you know, in this case, it would be increases, comma, decrease, comma, then something over here, comma. So go ahead, you'll have 1, 2, 3, 4, 5, 6, 7 things. And I'm going to give you a minute to do that. Get all of those fill in the blanks things in there, and then I'll show you the answers. Go ahead and do it, please. So guys, you're going to do all 6 of these, which is a much better way to do it. And I'm going to do when I do my next YouTube live, I'm going to sort of chunk these together. Okay, I'm starting to see some answers come in, and I'll show you the answer in a minute. Filling in all of this. Okay, okay, Amazing Arts, you got it. Other people are thinking. Keep going. You can totally do this. It's too blurry to read. Okay, and I just got a request to read this aloud because of a bad connection. So I got to check my settings on YouTube, which is making me very sad. YouTube is telling me that I've got a good connection. So Jojo, I'm wondering if that is your bad connection. But Jojo, I got you here. I'm going to read the whole thing aloud. P blanks tumor suppressor proteins, which would blank cell proliferation. Q indirectly inactivates X because X blanks cell proliferation. Inactivating X, which is what substance Q does, should blank cellular proliferation. Therefore, a combination of P and Q should lead to the blank amount of cellular proliferation, which is what is shown in the last bar of graph C. P and Q used separately should also blank cell proliferation, which is what is shown in the second and third bars of graph C. Absence of both substances would lead to a blank in cellular proliferation, which is what is shown in the first bar of C. Here's all the answers. No, here they are. Okay, so we've chosen the correct graph. You now have a sample justification. Anybody want to ask me any questions? You know, put some questions in the chat, and I'll see what I can do to clarify any things. Okay. All right. So, any additional questions? Hey guys, am I there again? Am I live? Okay, fantastic. I think you lost me for a minute, right? Oh, this is quite a new world, streaming. It was very nice when I could just be in the classroom with my students, though I have always, of course, enjoyed doing AT. Sarita, you're asking me how long is this live chat? I was thinking an hour. But I've only gone through one question. Okay, I'm back. Thank goodness. Okay. So let's go on and do something else. Okay, so guys, this was a goofy thing that I do with my own students. We have a class cheer where we go B-I-O-L-O-G-Y. That's our class's battle cry. And I thought that I would see how quickly people could type. It's actually quite challenging. B-I-O-L-O-G-Y in the chat. Let me see some B-I-O-L-O-G-Ys in the chats, and let's see it right now, okay? Just to know that you're with me. Let's go. I'm hearing some very nice feedback from Grant and Sky about some of my musical endeavors. Thank you very, very much. Amanda, there's no dashes. No dashes. The whole thing is to put the dashes in there. Who's got it? This was not what I expected. I'm seeing discrete letters coming up on the screen. Is that what happens? Oh, maybe this is an impossible task. Who knows? Ah, Chatticus did it. Amelia did it. Amanda did it. Yes, Amazing Arts. All right, fantastic. Okay, all right, so guys, that was good. Let's do a little fun activity, okay? It's a biology quiz, okay? You're gonna put your answers in the chat, all right? I'm seeing all these folks who have typed in B-I-O-L-O-G-Y, and they're gonna type something else. So I'm gonna show questions, and you're gonna type in your answers. All right, so everybody ready? Cell division that decreases chromosome number by half. What is it? I'm still seeing some B-I-O-L-O-G-Ys come in. That's fabulous. Okay. Yeah. It's coming in. Okay, fantastic. All right. Guys, you're all awesome. And the answer indeed was meiosis. All right, you guys ready? Next one. Okay, fantastic. Let's do another one. Where ATP synthase is found in plants. Okay, let's go. Come on, come on, come on, come on, come on. ATP synthase in plants, where's it found? Oh, I'm seeing some answers come in. Let's see some more. Come on. ATP synthase. Ah, wow. You know what? Sarita has by far the best answer. I'm wrong. What's going to be on the screen now is going to be wrong. In fact, I'm embarrassed to show it because I was just going to say chloroplast, but there's a better answer. It's the thylakoid membrane and the inner mitochondrial membrane because plants have mitochondria too. And who got that? Sarita, who said, was it Sarita who said both? It's the mitochondria and the thylakoid. And Mr. Glimmy said the cells, and that is really a great answer. Fantastic, Mr. Glimmy. Okay, it's the thylakoid membrane and the inner mitochondrial membrane. And again, thank you, Sarita, for being really the star of this because you identified that it was both. And I did what a lot of people do and I forgot that, you know, plants do cellular respiration too, just like we do in their mitochondria. So good for you. All right. What's the key enzyme involved in transcription? Key enzyme involved in transcription. Let's see it. All right, I'm waiting for some answers. Key enzyme in transcription. The enzyme that transcribes. Not seeing any answers come in. Yes! Ariana Rukin. You have to watch your language. Okay. Ooh, I probably shouldn't have said your last name, but it is there in the chat. Anyhow, so fantastic, guys. Indeed, it is RNA polymerase. That's the enzyme that transcribes. You guys ready for another one? Let's go. The only domain with an endomembrane system. Only domain with an endomembrane system. Very good. I'm seeing the answers come on in. Type it in the chat. Type it in the chat. Let's see. Fantastic. Fantastic. Okay, so indeed it is eukaryotes. You know, this is an interesting question. One of you just asked me if we need to know this. I think that it's certainly within the bounds of what the College Board could ask you on this year's AP exam about different structures in different types of cells. And the most fundamentally different types of cells are between, obviously eukaryotic cells and prokaryotic cells. And then within the prokaryotic cells, there's really two different domains that are really quite different from one another. If you got to Unit 7, evolution, the term prokaryote, it's a polyphyletic category. It's a bad evolutionary category because the domain that is archaea is actually not particularly closely related to the domain bacteria. And in fact, it looks like eukaryotes are more closely related to archaeans than we are to bacteria. And the origin of life and the emergence of the first cells, incredibly, incredibly complex and cool stuff. All right, so last question. All right, I did a YouTube song about this. In the Taylor Swift song, You Belong With Me, if Good Taylor, the kind of nerdy one, was the enzyme, and the boy across the street was the substrate, then Bad Taylor would be, she's the one with black hair, she's the one who's the cheer captain, stuff like that. Bad Taylor would be a what? Let's see some answers come in. This is a tough one. You guys know this song, right? Everybody knows this song. How can you not know this song? It's a big part of American culture. Ooh, I'm seeing some answers come in. I'm seeing something from Melissa. Okay, nice. Ben, Sarita, Ari, fantastic. Sky, Chaticus, Nissa, you guys are all awesome. Competitive inhibitor. I did a very goofy YouTube video about this a couple of weeks ago. A couple of weeks ago, it's actually a couple of months ago. Time flies during a pandemic, I guess, right? Grant, you don't know the song, everybody knows the song. You're on the phone with your girlfriend, she's upset. She's going off about something that you said. She doesn't get your humor like I do. That's the one. Anyhow, great song, one of my favorite songs. All right, so So it's already 11:50. I think we're just going to do one more of these. This one's a little bit easier, and then we're going to call it a day because I only anticipated that this would be an hour. So I'm going to read this aloud. So let's talk about what's going on here. Epinephrine, all right, also known as adrenaline, is produced by the adrenal glands. Those are right on top of your kidneys. The structural formula for epinephrine is shown at right. That's what this is over here. The two diagrams that follow, these two diagrams over here, are general models for signal reception and transduction. So part one, identify which model, model S or model T, best corresponds to the mechanism of epinephrine. Justify your response. Part two, epinephrine often has effects throughout the body. Some tissues are not affected. Explain. And then three, identify which model would bring about longer lasting effects. Justify your response. Okay? So, okay, so let's go on. I saw a couple of ambiguous comments, but I'm just going to go on. And so let's just take this piece by piece. Epinephrine, this is what we just told you right here. Epinephrine, also known as adrenaline, blah, 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 blah. So which model S or T corresponds to the mechanism of epinephrine? Justify your response. So let's do some analysis. Okay, so first of all, you guys are already jumping ahead. Don't worry about that. But let's just look at this molecule over here. So epinephrine, is it polar or nonpolar, and how do you know? All right, let's get it in the chat right now. Okay, I'm seeing some good answers. Okay, Amanda, you're asking a very good question. The answer is in the diagram itself. Ari, on target. Okay, let's get some more answers in there. Okay. Seeing a couple more answers come in. Oh, Mr. Glimmy, I'm glad I'm making you happy. That's making me very, very happy. Okay, so I'm looking for some justifications too. How do you know? How do you know that it's polar? Sky, nice. Chaticus, Hannah, want to add anything to your answer? Lizzie, how about you? Glimmy, you should know this. Okay, so I'm going to show you the answer, guys. Epinephrine is polar. And the reason why you know that is because these guys, this one, this one, this one, these are polar functional groups. Now, I can assure you that you're not going to need to know the name of functional groups on the AP exam on May 18th. But I also can assure you that it is possible that you might need to be able to identify a molecule as being polar or nonpolar and thereby know how it's going to behave in relationship to the membrane. So if you were able to take it to a place where you could say hydroxyl group, that's fantastic. But if you couldn't, then that's fine too. And as long as you knew that it was polar. Now, things that are polar, what's the story here? Because it's polar, epinephrine, what word would go through there? Blank, diffuse through the cell membrane. Okay, let's see some answers come in here. One word answer. Because it's polar, epinephrine, blank, diffuse through the cell membrane. What you got here? Nice. Fantastic. Okay, very good, very good, very good, very good, very good. Jojo. T-Bone is giving some positive feedback to Grant. That's fantastic. Amelia, Sarita. All right, fantastic. You guys are all got this, all right? So it can't diffuse through the cell membrane. All right, now, which diagram shows a ligand, a hormone that isn't diffusing through the membrane? You guys had this at the start. Just put in one letter. It's going to be so quick. Let's see the letters flow in. Whoa. Oh no. Let's go like this. Oh, I got it wrong. That's why I thought, okay, I knew I had a moment of panic there. So the answer should be T. I don't know what happened there. Little momentary lapse. Let's go like this. Okay. If you're my student, you know it's not the first time I've made a mistake. Yes, T is showing something that can't go through the membrane. Okay, now. All right, so now we've got that. We've got a justification. Epinephrine has effects throughout the body. Some tissues are not affected. Explain. Let's do a little bit of analysis. Epinephrine in model S binds with a membrane. What? What? Put the answer here. What is epinephrine binding with? Epinephrine's binding with that. What's the word for that? Cursor's actually blocking it right now. Okay, I'm seeing some of the answers come in. Let's see some more. The answer was, I'm sorry, I was really clear about this. Yes, so the answer for the previous one was T. I'm really sorry that I got it wrong in my PowerPoint. And it binds with a membrane receptor. So all of you put receptor, that's fantastic. And basically, here's the most important, it's not really the most important, but it's my favorite word in biology. If you're my student, you got to know this. The shape of that receptor is blank to the shape of the hormone. Let's see it. And guys, just so that you know, we're going to go a little over time. I'm not going to be able to end at 11 and finish this question, but it'll be shortly after 11. So, I mean, you know, I'm not going out anywhere. You're probably not going out anywhere either, right? Oh, I'm seeing the word come in. Fantastic. Okay. All right. So the word here is one of my favorite words in biology. It's complementary. All right. Let's do a little review. All right. Complementarity is one of the most important ideas in biology. And now let's talk about some other things that are complementary. Like, for example, enzymes and what's complementary to an enzyme? Okay, but enzymes, okay, so you got that, Alan, lock and key, but what's the key? All right, so the enzyme, I'm seeing it come in, it's just flowing in, fantastic. Amazing Arts, Alan, Amanda, Chaticus, you got this. Grant, Simi, Nisa, Melissa, everybody's got it, okay, beautiful. How about this one? Adenine, what's it complementary to? Let's go, come on, let's get it in there. Oh, this is making me so happy, all the people who are responding in the chat. That's fantastic. Damn. One of my students is pointing out that I made a spelling error. This should be complementary with an E over here. Oh, I gave the answer away. I didn't want to do that. Let me do this. That should be complementary there. I got to get a better pen. Everybody see that? Complementary is like when you say, hey, Mr. Wokenfeld, you're really doing a nice job. That's complementary. But, you know, when you talk about two things fitting together, that's complementary with an E. And people are getting it, adenine and thymine. Beautiful. All right, you guys are all over this. Codon. What's complementary to a codon? Georgia, that's very good. Very good. All right, I'm waiting for some answers to come in. And I see Krista. Krista, I made a mistake in the PowerPoint, and it actually is the model. Oh, multiple mistakes over here. This should also be a T. Thank you very much. Really sorry. Here, I'm going to try that silly pen thing again. Okay, how's that? It's pretty lame, but there we go. Okay, so codon, complementary to an anticodon. You guys are fantastic. Okay, and so just to answer this, the hormone only affects cells with a what kind of receptor? You guys know this. You've already typed it. Go ahead, type it in again. I'm just going to wait for one or two people to answer, and then I'm going to go on. Seeing some answers. No answers coming in yet. Hormone only affects cells with a what kind of receptor? Just one person, type it in, and then I'll know that someone's understanding me. Oh, good feedback from Naomi, and complementary, and that's right. Okay, so basically that's your justification for the answer. That's your response here. The hormone goes everywhere, but it only affects cells that have a complementary membrane receptor is really the main thing. And the sort of irony of this question is that epinephrine has very many widespread effects. It's the fight or flight hormone. It makes your pupils dilate. It makes the hairs on your, your arms and legs stand up. It makes your heart beat faster. It mobilizes glycogen to get broken down into glucose. But the main thing is that it, there are many types of cells that it doesn't affect, and that's really the main thing. Okay, so I think we're at the end of this question. Identify the model S or T that would bring about longer lasting effects. Justify your response. So here's model S, here's model T. Which one would have longer lasting effects? And just to bring this to a close, just without me saying anything, I want you to type in an S or a T into the chat and let me know what you think. Which one would have more permanent kinds of effects? And then I'll walk you through the whole thing. Seeing some answers coming in. Seeing some other answers coming in. All right. Alan's answered. Amanda has answered. Grant has answered. Lizzie, Sarita, Melissa. Okay, so I'm going to walk you through this whole thing in the interest of ending this. So, Model T is just changing cytoplasmic enzymes. If you remember to the work that we've done about like G-coupled receptors and stuff like that, you've got all of these enzymes inside the cell that are part of these signal transduction pathways. They are ready to go. That's what T is all about. In fact, you can sort of see how this diagram is indicating signal amplification right there. Now, on the other hand, S is showing a change in gene expression. In other words, what you see is that this hormone is actually entering into the cell, inside the cytoplasm, and it's not really clear, I guess it is shown, it gets picked up by a cytoplasmic receptor. That is usually a transcription factor that turns on expression of genes in DNA. So here we got DNA that's going to be made into messenger RNA, that was sort of represented in a funny way here. And that is going to what looks like is supposed to be a ribosome, but we're going to produce protein, all right? Now that is a slow process, and that is what steroid hormones are all about, and that's because steroids are nonpolar, and steroids can diffuse through the membrane, where they often bind with cytoplasmic receptors and then turn on genes. And just to sort of make this real to you, think about steroid hormones, all right? Now, it's been a long time since well, that's actually not true, but the main thing is that, you know, if you've been through puberty in the last 5 or 10 years, you know about what the effect of steroid hormones have been on your body. They produce long-term effects. The reason why there's a large degree of sexual dimorphism in our species, that means, you know, different phenotypes between males and females, is because of the influence of steroid hormones, which cause all of these secondary sexual characteristics. So, longer-term effects, this over here, short-term effects, enzymes right over here. And think about this, fight or flight, you got to have that happen fast. Long-term changes that are going to affect reproduction and things like that, that makes more sense to be happening with steroid hormone. So guys, just want to emphasize, we're going to bring this to a close right now. You can do this. You can totally do this. Our goal is to get up here, all right? And I know that every single one of you, if you put your time in, can get up to here where you're going to get your 4 or a 5 on the AP exam. We're not going to do that question now. I'm going to save that for our next chat. So I'm going to jump right to here, here. And I just want to say, here's a quote from the young Mr. W. There have been cell phones in 1979. I definitely would have used Mr. W's Biomania app to study for the AP exam. All right. So there you go. That's a quote from the young Mr. W. I want to totally encourage you. Free download. And then you do an in-app purchase to unlock all the content. It's totally worth it. And here's the old Mr. W. If you're not part of a class that's using science music videos, then you can buy your own subscription, and it's $19, and it's a fantastic investment in your education. So I thought I would end with a song. And I hope you sing along with me as I do this. Got more chords than I'm actually used to here. I think I can make this work. All right. So, you know, this is a time when, despite the fact that we're doing all of this physical isolation, social isolation, that's not something we should have. We should be leaning on one another more than ever. Sing it with me, all right? Sometimes in our lives, we all have pain. We all have sorrow. But if we are wise, we know that there's always tomorrow. Lean on me. When you're not strong, I'll be your friend. I'll help you carry on, for it won't be long till I'm gonna need somebody to lean on. Please swallow your pride. If I have things you need to borrow, for no one can fill all of your needs that you won't let show. You just call on me, brother, when you need a hand. We all need somebody to lean on. I just might have a problem that you understand. We all need somebody to lean on. Call I'll be around. Call me. Call me. And guys, I want you to know that that is true. I am there for you. If you need to email me about anything, there's a contact form that's on my website. Let me know about that. Again, I want to totally encourage you to, get the Science Music Videos subscription, buy the app. That's what you got to be doing. You got to be practicing with real questions. And I am going to sign off. I've got my next live chat like this scheduled for 4/14/20, 2 p.m. Pacific Standard Time. And I want you to encourage all your friends and stuff to join because, we're all in a tough spot. We're doing this distance learning thing. Distance learning to get ready for the AP exam, it's difficult. And so I want to be there. I want to support people. I want to do that through these live performances. And I want to do this through the app. So Naomi, I hope you can see it, that it's on April 14th. And Angelique is talking about Bill Withers, and he was such a legend, and I totally agree. And Ben, you are more than welcome. And I'm getting a goat that really makes my day. It took me a long time to figure out what that acronym actually meant. But so guys, you're the best. Take it easy. I'm going to sign off. I'm going to end this stream. Again, Science Music Videos, subscription, get the app. You're awesome. Let me know if I can do anything for you. And everybody be well. Bye-bye.